So thank you very much that I can present another cost project, which is, I think, closely connected to this one. It's also focusing on forest, but focus more on multi-taxon forest biodiversity. The chair of the project is Sabina Burrascanu from Rome, and I am the vice chair of it, and we are somewhere in the middle of the cost action. Some words about the background and the motivation and the aim of this project. Forest maintain a very high proportion of the biodiversity, which is true both globally and also for Europe. For the maintenance of this biodiversity, a crucial point is the sustainable forest management. In Europe, only the 1% of the European forest is in primary stage. Practically all European forests are managed nowadays or managed in the past. But there are a lot of effort for the conservation of forest biodiversity. If you see the conservation status of forest, about 80% of the European forests are unfavorable conservation status, and forestry is the key pressure for the maintenance of biodiversity. If we're looking which are the indicators of this sustainable forest management, we can see mainly landscape scale or standscape structural indicators, and we have very few indicators directly connected to forest biodiversity because it's a very hard and time-consuming job to analyze and collect direct biodiversity data. For that reason, we want to try a continental scale evaluation for the exploration between the relationship between the forest management and biodiversity, and we try to collect this kind of local studies from Europe. So the aim of the platform that we want to invite to a common network of the different researchers who has forest structure and multi-taxon biodiversity data. We want to make a network of forest monitoring sites, and based on this network, we would like to build a common platform and a common database in which we can make European scale evaluations. As an additional thing, we can share the protocols and the methodology of the sampling and analysis of different organism groups. And as a separate work package, we study separately the forest manipulation experiments when the dependent variables are multi-taxon biodiversity. Based on this platform, we would like to assessing the relationship between forest biodiversity structure and management in a European scale. We would like to develop and test the different sustainable forest management indicators and thresholds for biodiversity. And finally, we would like to make management guidelines for forest certifications and other purposes based on the direct data analysis. Similarly, like this cost action, we have about 30 European countries as members of the management committee. And during the work process, the first step was because we try to make a common platform, a common database that we have to agree the laws and the regulation, how can we use the data of this common data set. So we made a common shared bylaws. And after we have to harmonize the different protocols, so we collected the methodological descriptions and metadata of the different studies. So we have to make a harmonization for the different protocols. And we have to collect general information from the sampling units. And finally, we started to put together the raw data, including the forest structure and the data of different organism groups. This is the structure of our database that we have. The sampling units are the plots, which occurring in sites. The sites are larger areas when, in many cases, in different forest types, within one project, many plots. And sometimes we have to use the so-called stand scale, because in some project there are many plots within homogeneous stands, and in many organism groups the sampling was related to stand levels. So we have 3 hierarchical levels in the database. We have a separate database for the standing trees, including living and dead trees. We have a separate database for the lying deadwood, and we have a separate database for the species, including many organism groups. We are in the middle of this project, and the database is more or less ready. So there are 35 different projects involved from Europe in this common platform. We have more than 30 taxonomic groups and close to 4,000 sampling units in this database, of course with a lot of records, and you can see that it has a relatively high representation of different forest types of Europe. Some words about the representativity of our database. The blue columns are the proportion of different forest types in Europe, while the red show the frequency of the forest type in our database. So you can see that some forest types are overrepresented, especially beech forest and mesophytic deciduous forest, where some forest types are less represented in this multi-taxon project, like the boreal forest or the Mediterranean thermophilous forest. It was also very important that we try to minimize the missing values, especially on the site level or the plot level description. So it was a high effort that we should calculate some standardized variables like stand age, different silvicultural methods, which should be also standardized for a European platform. But now for this important explanatory variables are more or less filled. But for example, for deadwood, there are many projects which didn't collected data about deadwood. You can see that the different management types also well represented in the database. There are many shelter wood system, selection system, unmanaged forest, and different type of clear cutting or coppices. It is also possible that in many forest types, the different management systems are well represented. So we also have a possibility to compare the management systems within one forest type. And this is the distribution of organism groups. In average, close to 5 taxons are collected in each plot. So these are a really multi-taxon data set. The minimum level was 3. There are 2 or 5, 1 or 2 because of the stand, because for some organism groups was collected in some project on stand level. You can see in this graph that which are the most representative organism groups in this data set. We have 6 organism groups which are quite common, like vascular plants, bryophytes, lichens, beetles, and fungi. One of the first output of the project that we summarized the different protocols, and we give a description for the most common and most efficient sampling protocols for different organism groups. Okay. We made some kind of standardization on the database. For example, the species richness was standardized for the site level, maximum species richness, and so on. And there are some description about the structure that most of the forests are that we have many relatively young forest stands, and most of the forests based on the basal area are relatively closed. I would like to mention also the experiments. It is a separate database. Here the experiments don't make a common database, they only share the protocols and the metadata. There are 27 experiments in Europe, and there is some forest structure manipulation and biodiversity response. Most common in this manipulation are the different type of cuttings, like gap cutting, thinning, clear cutting. There are many microhabitat manipulation, deadwood accumulation, microhabitat manipulation, some game closure, and so on. And finally, I would like to some words about how would like to use this relatively big database for the future. So there are target aims and deliverables of the project, but besides, we are open for many questions or research papers, and there is a consortium who decide that some other project could use this common database, and all custodians and data owners should participate on it. Here you can see that we have some ongoing works or little project based on this data set. And we also use in the cost the other possibilities like the short-term scientific mission, when somebody is going to a partner and doing some short-term intensive work for the cost action, or the virtual mobility grants, which is the same, but you stay at home and working by a home office way. We have also training schools in the project and conference grants. And if you are interested to the results, we will have a symposium in August here in Prague in the SECB conference. You can find the details in the website of the project, and also in the Twitter and other media platforms. And this is the first kickoff meeting of the project. Here is Sabune Burascano, the leader of the chair of the action. And I hope that probably in the future we have possibilities to collaboration between these two actions. Thank you very much for your attention.
Standard definition about multi-taxon biodiversity?
Yeah, we defined it that a project can include this platform if they are collecting minimum 3 organism groups, and within these organism groups should be also animals. So the classical vegetation inventory is when you only inventory the plants and probably the lichens. It's not a real multi-taxon. So the multi-taxon project, when you are studying in one plot in the same times, minimum 3 functionally different organism groups.
Okay, I think that is all. If there is no question, thank you, Péter.
Thank you very much.